When we think about how a species goes extinct, our minds usually jump to the blockbuster movie scenarios. We picture a massive meteor striking the Earth, a bulldozer relentlessly clearing a vibrant rainforest, or the sharp crack of a poacher's rifle echoing across the African savanna. But the truth is, for many of the world's most endangered animals, the real battle for survival isn't happening out in the open. It is being waged invisibly, at the microscopic level, deep within their DNA. Long after the poachers have been locked up and the habitats have been fenced off and protected, a silent, creeping threat remains. This threat is known as a genetic bottleneck. It is a severe, dramatic reduction in a population size that essentially strips a species of its genetic diversity, leaving the survivors incredibly vulnerable to disease, sudden environmental changes, and the slow, insidious decay of inbreeding. Welcome to Fun Facts Online. Today we are diving deep into the microscopic battlefields of conservation, exploring how endangered species are rebounding from the very brink of oblivion, and how scientists are using everything from cross-country translocations to advanced cloning and synthetic biology to rescue the genetic legacy of our planet. To truly understand a genetic bottleneck, you have to imagine a species' genome as a vast, ancient library. This library contains millions of books, and these books represent the genetic diversity, the various alleles and traits that the species has accumulated over hundreds of thousands of years of evolution. This diversity is the ultimate survival toolkit. If a new disease emerges, some individuals will hopefully possess the specific book containing the immune response needed to survive it. If the global climate shifts and warms, some will have the genetic instructions to tolerate that heat. But when a population undergoes a sudden and drastic decline, it is as if that ancient library catches fire. Imagine a population of 100,000 animals reduced to just 20. The vast majority of those genetic books are burned forever. Even if dedicated conservationists step in, protect those 20 survivors, and help them multiply back into a population of thousands, there is a catch. That entire new population is built exclusively from the tiny surviving section of the library. The sheer numbers of animals might have recovered, but the genetic information is permanently lost. This catastrophic loss triggers a dangerous phenomenon known as genetic drift. In this state, random chance events can cause the few remaining genetic variations to fluctuate wildly or disappear altogether. Even worse, the surviving animals eventually have no choice but to mate with their close relatives. This inbreeding increases the likelihood of homozygosity. That is a biological state where an individual inherits identical copies of a specific gene from both parents. Now, in a large, diverse population, many harmful disease-causing mutations are recessive, meaning they remain hidden as long as they are paired with a healthy, dominant gene. But when relatives mate, these hidden, dangerous mutations suddenly find themselves paired up. They step out of the shadows and express themselves as physical deformities, lowered fertility, and severely compromised immune systems. This devastating spiral is called inbreeding depression, and it can pull a species into an extinction vortex. Poor health leads to fewer offspring, which leads to even tighter inbreeding, until the entire species simply collapses. Yet, nature has a remarkable way of defying these odds. And where nature struggles, modern science has stepped in to lend a powerful hand. To see the long-term, echoing scars of a genetic bottleneck, you don't need to look at a species struggling in a zoo today. You only need to look at the fastest land mammal on Earth. The cheetah is a marvel of biomechanical engineering. It can reach highway speeds in a matter of seconds. But beneath that sleek, athletic exterior lies a genome deeply scarred by ancient traumas. Unlike many species facing genetic crises right now, the cheetah's genetic uniformity is not primarily a modern disaster caused by humans. Scientists have discovered that cheetahs actually suffered two massive historical bottlenecks long before the invention of the rifle or the combustion engine. The first one occurred roughly 100,000 years ago. Cheetahs originally descended from ancestors shared with the American puma. As they expanded their range out of the Americas, crossing the Bering Strait into Asia, Europe, and eventually Africa, these migrating populations dispersed in very small groups. This rapid expansion over vast, scattered territories heavily restricted their gene flow. But the second, much more devastating bottleneck took place between 10,000 and 12,000 years ago. This coincided with the end of the last ice age. a brutal mass extinction event that wiped out many of the world's large vertebrates. The North American and European cheetahs were completely eradicated. All that was left was a tiny handful of survivors in Africa and Asia. From this unbelievably microscopic genetic pool, all modern cheetahs are descended. The result of this ancient catastrophe is a species that is, essentially, a clone of itself. Back in the 1980s, researchers made a startling discovery. Cheetahs possess an incredibly uniform major histocompatibility complex, or MHC. This is the set of genes responsible for the immune system's ability to recognize foreign invaders. The uniformity is so extreme that a cheetah will actually accept a skin graft from an entirely unrelated cheetah without its body rejecting the tissue. Biologically speaking, unrelated cheetahs behave like identical twins. But this extreme lack of genetic variation comes with a terrifyingly heavy price. Today, cheetah populations are perilously vulnerable to diseases like feline coronavirus, and they suffer from severely compromised reproductive systems. Studies have shown that between 70 and 80% of cheetah spermatozoa are malformed, compared to just 30% in domestic cats. While global populations currently hover around 6,500 to 7,100 individuals, their genetic fragility means that a single, highly contagious novel pathogen could theoretically wipe out the entire species overnight. To fight this, conservationists have to meticulously manage captive breeding programs, using advanced stud books to ensure the widest possible genetic spread among the remaining animals. Furthermore, facilities like the Cheetah Conservation Fund have established biobanks, freezing hundreds of sperm and tissue samples to preserve whatever diversity is left. Recently, breakthroughs in vitro fertilization have allowed scientists to produce cheetah embryos and successfully transfer them to surrogates, buying the species vital time. The cheetah is living proof that a species can survive a near-total genetic wipeout, provided it is given enough time and space to stabilize. But what happens when the bottleneck isn't an ancient relic, but a modern tragedy unfolding right before our eyes? Enter the Florida panther. The Florida panther's crisis is one of the most successful, yet incredibly complex, human interventions in the history of conservation. This subspecies of the widespread puma once roamed across the entire southeastern United States. However, aggressive, unregulated hunting and rapid habitat destruction slowly boxed the cats into less than 5% of their original territory, isolating them in the remote swamps of South Florida. By the 1990s, the situation had become catastrophic. The wild population had plummeted to fewer than 30 individuals. The effects of the genetic bottleneck were highly visible, and they were gruesome. The panthers were suffering from severe inbreeding depression. Males exhibited cryptorchidism, which is a condition involving undescended testicles that leads to sterility. Congenital heart defects were rampant across the population, and many of the animals sported distinctive, highly unusual kinked tails and weird cowlicks of fur on their backs. Biologists ran the numbers and predicted that without immediate, drastic intervention, the Florida panther would be completely extinct within a century. So, in 1995, conservationists made a highly controversial decision. To introduce fresh DNA and violently break the cycle of inbreeding, they translocated 8 female pumas from West Texas directly into the Everglades. The logic behind this bold move was grounded in natural history. Before human expansion fragmented their habitats, Texas pumas and Florida panthers would have naturally encountered each other and interbred at the edges of their respective territories. This strategy, known as genetic rescue, worked almost miraculously. The introduction of the Texas females rapidly reduced the occurrence of debilitating inbreeding traits, massively bolstering survival and reproductive rates. 30 years later, the Florida panther population has rebounded tenfold, rising to an estimated 200 individuals today. However, genetic rescue is not a magic wand, and it comes with deep anxieties. One of the major concerns at the time was genetic swamping. Scientists feared that the unique genetic ancestry that made the Florida panther a distinct subspecies perfectly adapted to the swamps would be completely overridden and erased by the influx of Texas DNA. But a landmark study published in the Proceedings of the National Academy of Sciences in July 2025 finally put these fears to rest. Researchers from UCLA, UC Berkeley, and the Florida Fish and Wildlife Conservation Commission sequenced the genomes of the post-translocation panthers. What they found was absolutely fascinating. While the rescue successfully decreased harmful homozygosity and increased overall genetic diversity, the original local ancestry of the Florida panther remained highly prevalent. Natural selection had stepped in, favoring the retention of traits suited to the harsh Florida swamps, while utilizing the new Texas DNA simply to mask the harmful recessive mutations. It was a perfect biological compromise. Yet, that same 2025 study offered a very sobering caveat. While the total number of expressed harmful mutations dropped significantly, those dangerous alleles were not entirely purged from the gene pool. They were simply hidden in a heterozygous state, meaning they were paired with a healthy, dominant gene from the Texas line. The researchers warned that the benefits of the 1995 genetic rescue might only be transient. Because the population remains geographically boxed in by highways and human development, inbreeding will inevitably rise again. This means the Florida panther is now a conservation-reliant species. It will require ongoing, permanent human management and likely future genetic infusions just to survive. If the Florida panther required a genetic rescue, our next animal required a full biological resurrection. The story of the black-footed ferret, North America's only native ferret, pushes the very boundaries of modern science, taking us from the absolute brink of extinction into the futuristic realm of synthetic biology and cloning. The black-footed ferret is a slender, highly specialized predator that depends almost entirely on prairie dogs for both food and the burrows they use for shelter. Sadly, they became severe collateral damage in the 20th century. As farmers and ranchers systematically poisoned and exterminated prairie dog colonies across the American West to protect their agricultural lands, the ferret population crashed right alongside them. By the mid-1970s, the species was presumed to be globally extinct. Not a single one could be found. Then, in 1981, a miraculous accident occurred. A ranch dog in Meeteetse, Wyoming, dropped a freshly killed animal on its owner's porch. Biologists were stunned to realize it was a black-footed ferret. This chance discovery led them to a tiny, hidden relict population. But the joy was short-lived, as disease soon swept through the remaining colony, killing them off one by one. In an absolute last-ditch effort to save the species from blinking out of existence, the United States Fish and Wildlife Service captured the remaining 18 wild ferrets to start a captive breeding program. But the bottleneck was tighter than anyone feared. Out of those 18, only 7 individuals successfully bred and passed on their genes. Think about that. Every single black-footed ferret alive for the next 3 decades was descended from just those 7 founders. It was an extraordinarily tight genetic bottleneck. As the captive population was slowly reintroduced to the wild, growing to around 400 to 500 individuals, their profound lack of genetic diversity left them highly susceptible to diseases, particularly the devastating sylvatic plague. But the conservationists had a secret weapon, hidden away in deep freeze. It was called the frozen zoo. Back in the 1980s, visionary scientists at the San Diego Zoo Wildlife Alliance had the incredible foresight to cryopreserve the living cells of several ferrets. One of these was a female named Willa, who had died in 1988 without ever contributing offspring to the captive population. Because Willa was entirely genetically distinct from the 7 original founders, her frozen cells contained lost variations of the black-footed ferret genome. Decades later, a biodiversity nonprofit named Revive and Restore partnered with a cloning company called Viagen Pets, along with government agencies, to attempt the seemingly impossible. Using a technique called interspecies somatic cell nuclear transfer, scientists took Willa's frozen genetic material, carefully injected it into an empty egg cell from a domestic ferret, and implanted the resulting embryo into a domestic ferret surrogate. On December 10, 2020, history was made. Elizabeth Ann was born. She was the first ever clone of a native United States endangered species. Elizabeth Ann proved that cloning was a truly viable tool to reach back in time and shatter a genetic bottleneck. But her journey wasn't perfectly smooth. As she matured, Elizabeth Ann developed a condition known as hydrometria, which is fluid retention in the uterus, along with an underdeveloped uterine horn. This rendered her unable to breed. Now, because these conditions are relatively common in ferrets in general, they were not attributed to the cloning process itself, but it did temporarily stall the hopes of passing her vital 35-year-old DNA back into the living gene pool. But scientists did not give up. Knowing the core technique worked perfectly, they simply repeated it. In May 2023, 2 more clones from Willis' original cell line, named Antonia and Noreen, were born. And then, in 2024, the massive breakthrough finally arrived. Antonia successfully mated and gave birth to 3 healthy kits. This marked the very first time in human history that a clone carrying previously lost genetics successfully contributed offspring to an endangered population. The success rapidly cascaded. By the summer of 2025, 4 more litters, totaling 12 new kits, were born to Antonia, Noreen, and Antonia's offspring. Willa, a ferret who had been dead for over 35 years, officially became the 8th founder of her species. By breaking that stubborn 7-founder bottleneck, scientists artificially restored genetic diversity that nature had completely erased. And they aren't stopping there. Researchers are currently exploring the use of CRISPR gene editing technology as a complementary tool. They hope to literally tweak the ferret DNA to make them innately resistant to the sylvatic plague. The black-footed ferret has shifted from a grim cautionary tale of extinction into an incredible frontier of what scientists are calling intended consequences, proving that human biotechnology can actively heal the genetic fabric of the wild. We can see similar struggles and high-tech interventions when we look up at the skies. The California condor, North America's largest land bird, provides a stark visualization of how a bottleneck permanently truncates a species' evolutionary history. Due to severe lead poisoning from scavenging hunted carcasses, direct hunting, and massive habitat destruction, the California condor population plunged to an agonizingly low 22 individuals by 1982. In a dramatic and highly controversial move, conservationists made the choice to capture every single remaining wild bird. The entire modern population, which now thankfully exceeds 500 individuals, is descended from just 14 genetic founders representing 3 specific genetic clans. But what did they lose to get here? To understand the true cost of this survival, scientists recently utilized a field called museomics. This involves extracting ancient DNA from historical museum specimens dating back centuries. The findings were absolutely staggering. This ancient DNA analysis revealed that prior to the bottleneck, the historical condor population possessed a minimum of 18 different mitochondrial DNA haplotypes, but the 14 founders of the captive program brought only 3 haplotypes with them. In biological terms, the California condor lost more than 80% of its maternal genetic lineages in just a matter of decades. While the birds have been physically saved from total extinction, their genomic erosion leaves them navigating a rapidly changing modern environment with only a fraction of their ancestral evolutionary tools. This kind of genomic erosion is incredibly common in island species, which are inherently prone to bottlenecks due to their geographic isolation. Take the Seychelles magpie robin, an endangered passerine bird. It faced a massive decline that perfectly coincided with human settlement of the islands about 250 years ago. The population cratered to a mere 20 to 30 individuals. Through intense conservation translocations, aggressive habitat restoration, and the provision of medical care and supplementary feeding, the population has bounded back to approximately 500 individuals spread across 5 islands. However, computer simulations analyzing their genome have highlighted the immense value of continued translocations. Conservationists literally have to keep moving birds between the islands to enact continuous genetic rescue, halting the ongoing genomic erosion caused by island inbreeding. It is a never-ending game of biological musical chairs, just to keep the species viable. Now, given all these devastating consequences of genetic bottlenecks, you might be wondering, how do some species experience severe bottlenecks and yet emerge seemingly completely unscathed? This brings us to a fascinating, somewhat paradoxical evolutionary phenomenon known as purging. Let's look at the northern elephant seal. In the late 19th century, commercial hunting for their incredibly valuable blubber decimated the species. By the 1890s, the global population of northern elephant seals was reduced to an estimated 20 to 100 individuals, entirely isolated on a single island off the coast of Mexico. From this incredibly tight bottleneck, the species was finally protected from hunting, and their numbers absolutely exploded. Today, there are hundreds of thousands of northern elephant seals dominating the Pacific coastline. Despite undergoing a bottleneck, just as tight as the Florida panthers or the black-footed ferrets. The elephant seals show almost zero signs of inbreeding depression. They don't have kinked tails. They don't have mass fertility issues or heart defects. So, why did they survive so flawlessly? The answer lies in the speed and the absolute severity of the bottleneck, combined with the historic genetic load of the species. You see, when populations decline slowly over thousands of years, slightly harmful recessive traits have time to drift and accumulate. But when a bottleneck happens instantly, such as a mass slaughter over just a few decades, the resulting extreme inbreeding happens incredibly quickly. The lethal and severely detrimental mutations are abruptly exposed in the homozygous state. The individuals carrying these twin defective genes simply die before they can ever reproduce. This brutal process rapidly purges the worst of the genetic load straight out of the gene pool. The survivors of the northern elephant seal slaughter just happen to be relatively free of deleterious alleles to begin with. Thus, when they rebuilt their massive population, they were essentially building from a genetically narrow but fundamentally very healthy set of blueprints. However, purging is an incredibly dangerous gamble. It only works if the species has a very high reproductive rate and a relatively clean gene pool from the start. And even when it works, it usually results in an absolute loss of adaptive diversity. Elephant seals might be thriving today, but their uniform genetics mean that if a novel respiratory virus uniquely adapted to marine mammals sweeps through the Pacific Ocean, the entire population could be catastrophically vulnerable. They survive the bottleneck, but the invisible sword of genetic uniformity still hangs precariously over their heads. As we move deeper into the 21st century, the fight to overcome these genetic bottlenecks has evolved from a purely ecological discipline into a cutting-edge molecular science. We are no longer limited to just shuffling the remaining deck of living animals. We are quite literally learning how to print new cards. Biobanking is the absolute foundation of this new era. Facilities like the frozen zoo act as biological time machines, securely storing living cells, sperm, eggs, and embryos of thousands of threatened species at temperatures of -196 degrees Celsius. These repositories ensure that even if an animal dies without ever reproducing, its unique genetic diversity is not permanently lost to the species. The successful cloning of Elizabeth Ann, Antonia, and Noreen from 35-year-old frozen cells is the ultimate proof of concept that extinction is no longer necessarily permanent, as long as the DNA itself is preserved. Looking forward, technologies like CRISPR-Cas9 offer the tantalizing possibility of highly targeted genetic rescue. If a bottleneck has stripped a species of its disease resistance, scientists could theoretically just edit those defensive genes back into the population. For the black-footed ferret, researchers are already investigating how to edit immunity to the sylvatic plague directly into their genome. For other species facing the brutal realities of climate change, genes granting higher heat tolerance or better water retention could be synthesized and safely introduced. This represents a profound paradigm shift in conservation biology. For decades, the prevailing philosophy was strictly one of preservation, simply fencing off habitats, minimizing human interference, and letting nature take its course. Today, conservationists acknowledge the power of intended consequences. As the Revive and Restore team noted regarding the ferret cloning milestones, inaction and a fear of the unknown often guarantee the extinction of a bottlenecked species. By actively intervening with advanced biotechnology, we can deliberately restore genetic diversity rather than just accepting the bottleneck as an inevitable death sentence. A genetic bottleneck is a stark, sobering reminder of the absolute fragility of life. It demonstrates that a species' survival is dictated not just by the abundance of its physical habitat, but by the microscopic, invisible variability woven deeply into its cells. When a species loses this diversity, whether through ancient glacial shifts like the cheetah or modern human encroachment like the Florida panther, it loses the vital flexibility to navigate a rapidly changing world. Yet, the remarkable rebounds of these species are equally a testament to extraordinary resilience. Nature possesses an astonishing capacity to persist, to purge its own weaknesses, and to multiply against all conceivable odds. And where nature hits its absolute limit, human ingenuity has confidently stepped into the breach. We are the species that forced so many others through the terrifying crucible of a genetic bottleneck. But we are also the only species capable of understanding the double helix, sequencing vast genomes, tracking alleles, and using cloning and genetic rescue to actively undo our own damage. The incredible recovery of the Florida panther, the sci-fi cloning of the black-footed ferret, and the intensive, relentless management of the California condor show us that extinction is not always a one-way street. By combining traditional ecological conservation with cutting-edge genetics, we are not just saving individual animals. We are actively securing the ancient, complex, and beautiful library of life for generations to come. Thank you for tuning in to G Fun Facts Online. Keep exploring and stay curious.